Apologies for the technical challenges, everybody. This is Stephen Burley, Director of the Institute for Quantitative Biomedicine, welcoming you to the first of our fall 2023 seminars. I want to make a few announcements before Jason Kelber introduces today's speaker. Here is the lineup for the fall. Please join us next on the 11th of October, when we'll be hosting Professor Squire Booker from Penn State University. I want to congratulate Jennifer Jiang on receipt of the 2023 IQB Inclusive Leadership Award in recognition for her outstanding achievements. Thank you for everything you do, Jennifer. And urge that there be nominations, either self or from others, for the 2024 IQB Inclusive Leadership Award. The due date is October the 31st. All the details can be seen on this slide, and you can get further information from Michelle Sangera, whose email is provided in the bottom right of this slide. I want to take this opportunity to announce some upcoming crash courses, virtual crash courses, on the use of the RCSB Protein Data Bank APIs, application programming interfaces, for bioinformatics analyses and machine learning. The first of these will take place on October the 12th and another on October the 19th. And for night owls and those who are in Asia, there'll be a session. The same session will be presented part 2. We've presented a second time on the 27th of October. I also want to draw your attention to our 2024 winter session boot camp on science communication. Applications are due by email on October 31st, and the details can be, and together with the application materials, can be found at the URL shown at the bottom of this slide. There are opportunities at RCSB Protein Data Bank here in the building for various roles. We're looking for a database administrator at Rutgers, also a postdoc expert in metalloproteins and organometallic chemistry. There's also an opportunity at with our team at UCSD for a postdoc trained in bioinformatics. We have gap year opportunities here at Rutgers, and we also have undergraduate summer research opportunities. And I urge all of you to consider joining the RCSB Protein Data Bank and learning how to play poker at a very high level. I also want to draw your attention to an ASBMB workshop that the Protein Data Bank will be putting on October the 19th from 2 to 3:30 p.m. Eastern. It's a members-only event with free registration. So if you're not already a member of ASBMB and you want to attend, please join this worthy organization. The title for the workshop is Teaching Enzymology with the Protein Data Bank and the subtitle is From Pandemic to Paxlovid. So with that, I'm going to stop sharing and turn the proceedings over to Jason Kelber in the room to introduce our speaker for today from Thermo Fisher. My thanks to the Thermo Fisher team for visiting Rutgers, New Brunswick today, and for the great products that you and your team make.
Thank you, Stephen. While we switch over to the slides, I will introduce our speaker. Thank you all for attending. So we're very happy to be hosting actually quite a few folks from Thermo Fisher up today. Thermo Fisher has been a really valuable partner for the Protein Data Bank and for our research here at Rutgers. And we'd like to host Geoff Perumal, who's going to be telling us today about Cryo ET workflows, which is a topic that is near and dear to some of the folks in this room trying to implement some of these cutting-edge techniques. So Geoff's background, Geoff actually has 10 years of experience in an academic setting as an electron microscopist. And from there, he went to the microscope manufacturer side of things. Is this on? Okay, I'll switch microphones. Where he's worked at 2 of the 4 major focused ion beam and SEM manufacturers, Carl Zeiss, which is where we first engaged with him at Rutgers, and now at Thermo Fisher. So I'm very interested to hear what he has to tell us today. But before that, in a small thanks of y'all's partnership in structural biology, I'd like to give you guys some protein data bank calendars so you can see your molecules every month. Thank you guys. Yeah, so turn it over to you, Geoff. The floor is yours.
Okay, so if you all bear with me 1 second here, I'm just going to switch this over. All right, so perfect. So thank you all for your time today. My name is Geoff Perumal, and I serve as the North American product specialist for our dual beam and traditional SEMs, cryo and room temperature as well. So as Jason pointed out, I've been in industry for about 7 years, working on a lot of the workflows which we'll discuss today. So as for today, we're going to talk about electron tomography, how it's being utilized, how it's a little bit of the history of it, and then we'll dive into a couple of advances which I see as improvements in efficiency as well as yield in these resultant lamellae. And we'll define lamellae for those of you who are newer to tomography as we move along here. So I'm breaking this up into 2 sections. Again, a little reintroduction to tomography, as well as some of the rationale behind why I see it as such a huge part as a driver of science. And then we'll go into these 2 techniques, those being the integration of fluorescence for cells grown on grids, and then what comes next when we want to take the cells to the tissue and how we approach that for tomography. So I think when we talk about all these techniques, it's kind of good to put them all together, understand how they relate in terms of scale as well as resolution. From our simple light microscopes, which range of broad size from microns to millimeters, all the way down to approaching those hundreds of nanometers, all the way down to our X-ray crystallography, which we then really cut our sample size but improve resolution down to the single angstroms range resolution. There's all these techniques in between: cryo electron microscopy, confocal microscopy, standard TEM. They all fit in, and each comes with a cost, if you will. As you improve your resolution, typically you down your sample size. What makes cryo electron tomography such a neat inclusion, as I back up to the slide I was describing, is that it kind of goes through the range of these sizes. So electron tomography can get into these tens of microns in sample size, but we don't always necessarily sacrifice our resolution. These techniques using subtomogram averaging can get down into the single angstroms range, probably in that 3 to 4 angstroms is where we comfortably see it. I've seen some ribosomal data, which we'll talk about, which got down to a 2.5 angstrom. So approaching that structural level where we could really start to answer those questions. This is one of 3 techniques, which is typically done with a cryo TEM. The other 2 being what really drove a lot of this, the single particle analysis, where we're taking individual proteins, putting them down on grid, doing some back-end work to get our single angstroms resolution. The X-ray crystallography is another one of these hallmark techniques with it. Right, the latest technique, obviously, is cryo electron tomography. What I think is really neat about it is, as opposed to a lot of other scientific directions, cryo electron tomography has kind of worked backwards in microscopy. Microscopy started out with wide field, we then went to confocal, super resolution, and it's always been this better resolution approach. Tomography actually kind of took the opposite angle, where we started with SPA and these single angstrom resolutions, and that's great to understand your protein, its folds, what's happening there, but it's a protein on a grid. So what does it look like when we put it in a cell? So now we're talking about a bigger piece of material, in this case, a cell. And we'll talk about how we actually backed it up then, because now we know what's happening on cells. Well, what's it look like in the animal of interest? And that's going to be this tissue approach, which is this lift out part, which we'll talk about third. Let's advance here. So cryo electron tomography is a process of taking a cell or tissue and using an ion beam to thin down the material above and below it to about 200 nanometers. That resultant lamella, which again we'll define in a few slides, is brought into a cryo TEM where we're doing some tilting of a region of interest and then back-end sub-tomogram averaging to get highest resolutions. But what I think is also really neat about it is it also provides some context. We're not just looking at our individual protein. We start to get a feel with these, coupled with these 300 kV cryo systems, about what's happening in context. So I always like to start with this one because it's a great, beautiful, first off, back-end projection, or excuse me, back-end imaging. But what we're seeing here is all of our organelles inside of our cell. So Golgi, cytoplasm, ribosomes are taking advantage of these great high-resolution techniques to really start to understand what's not just happening in our area of interest, but more in the broader context. Now, keep in mind that these techniques are starting to be implemented by a lot of labs, and we're seeing it, in a lot of these labs which feature these high-end cryo TEMs, these cryoses and glacioses. We're at a point of about 44% of our labs are now incorporating cryotomography, a very nice upward projection. And as you would imagine with that, we're seeing a lot more high-end publications which focus on cryo electron tomography. So here what we're looking at in this lighter blue was the trajectory of single particle, and it was a big takeoff with the advancements of cameras to really help support it. And we're seeing now in this darker blue line here, a similar trend happening with tomography. And I think we're going to see this continue to hit a trajectory such as SPA with some of these bigger improvements with internal fluorescence, lift out, where it really starts to get adopted to what's happening inside our cells, what's happening inside of our animals. So cryo-electron tomography, for all intents and purposes, happens inside your cryo TEM. That's where the tilting occurs. That's where we get all our data and then the resultant back-end curation and segmentation. But I always like to think of cryo-electron tomography as a workflow that begins the second you freeze your samples, whether it's, again, cells or tissues, through an intermediate, which is a FIB, which is a lot of what I spend my time doing, which will allow us to target areas inside of cells, thin down areas to get this resultant lamella. And then from there, we bring it into our cryo TEM and continue that workflow through the ultimate sub-tomogram averaging. So lamella, I've used that term a lot. I think it's good to kind of define it. Lamella is basically that resultant thin down process from our FIB beam. So we start out with a group of cells or tissues which are quick frozen. They're quite thick in the magnitude of tens of microns to hundreds of microns, depending on our methodology. That in itself, as we've seen in a couple of slides, is way too thick for a 300 kV beam to penetrate through. So the intermediate process of thinning down will occur inside of a FIB, resulting in these lamellae, which, as you can see here on a SEM, is still a little bit too thick to be translucent. And our cryo TEM, we could easily penetrate it. Once we're able to see on the other side, we're now able to start to do that tilted tomography in this workflow. These lamellae, as you saw in the previous image, and hopefully this movie plays here, are capable of two things as I see it: giving you context as to what's happening in a broader area. So this is a similar movie to what I showed before, where we now start to understand the interaction of our ribosomes and our Golgi. But the other end, the side which might interest a lot of our SPA folk, is also true as well. Let's see if I can get this one to play, where we're also able to take some of these finer particles, in this case, a ribosome here inside of a thin down lamella, and yield quite high resolution. So this is an example of a ribosome at 4.5. More recent works have shown this ribosomes down to 2.5 through tomography as well. So really approaching some of those highest resolutions that SPA has offered up as well. So 30 seconds on a FIB, just to make sure we're on the same page here. Focused ion beam is SEM is very similar to a traditional EM in the sense that we have a primary E-beam, which comes down our column. How it differentiates itself from a traditional SEM or a TEM is that There's a second beam inside the tool, which is our focused ion beam. I might refer to as an I-beam, which will actually do the ablation of your sample. So we're basically viewing our sample and removing, viewing, removing, till we get that resultant 200-ish nanometer lamella. The process basically now is we'll take cells. So for cells, that is cells are quick frozen into ethane. This process will prevent any devitrification of our samples. So the ice is pure ice. We do a little bit of blotting here to remove any excess there as well. These samples are transferred inside the aforementioned fib, put into a specialized holder. A quick coating is done of a heavy metal, of platinum typically, to allow the fib beam to behave most properly and mill evenly, but of also equal importance to prevent any additional charge. Presuming a lot of us are electron microscopy people in here, but for the light microscopists, charge is the EM equivalent to noise. So we want to get a nice clean image. Once that's done, then that process of iterative ablation and imaging can occur. A cell is identified. Softwares are basically now brought in to identify the region within our cell if we're able to do so, creating these two windows above and below your area, and then the ablation occurs. And we'll just let this one walk through here. And what we'll see ultimately is that fib will remove everything, including the grid below it, and then everything on north of it. Our platinum layer will remain behind, again, to help control our charge. So why are we doing this? I think it's kind of also worthwhile to go back and dive back here. I don't know if the colors are coming out here on your end. Oh, sure, thanks. But a lot of the interesting biology for tomography occurs right around the nucleus, which, as I pointed out before, is just too thick for an E-beam to penetrate itself. Peripheral work on our cells are certainly good enough to do some just regular tilting without a fib. But when we get to where a lot of that interesting biology happens, where our Golgi is, where our nucleus is living, we need to include this intermediate. This is a nice little depiction of what happens on a tilting of a cell that has not been fib milled. Again, our periphery is thin enough for the E-beam to penetrate. But once we get to all the interesting place around our nucleus where the biology happens, it becomes quite opaque quite fast, meaning that we're gonna not be able to actually see what's happening there. So these thin down lamellae are brought into our cryo TEMs, where I'm sorry if I keep doing this, but our tilting does occur. Our sample, an ROI is selected, and then that same area is imaged at multiple tilts, about 3-degree increments, plus or minus 70 degrees, if we have that clearance. And then the back end image is aligned, and you could do these segmentations, our subtomogram average. And here's just yet another example of this. So this is a lot of the early advancements that occurred in cryo electron tomography. It was a really wonderful approach to get inside the cells, as we've pointed out, but that kind of begs the question, and to the more broader audience, to those cell biologists, of, well, what happens if we want to target a specific area within our cells? Now, that's one of these first advances, which I'm going to kind of dive into, and that's the idea of internal fluorescence. So this movie, which should be playing in just a second here, is something which we just saw, so I'm not going to re-describe it, but this was the approach that a lot of early cryo electron tomography took, was let's just cut into a cell and basically hope that what we want to see is there. And there was a couple of approaches which were taken, one which I call a spray and pray. Let's go ahead and just cut a lot of cells and use the law of numbers to say that one of them is going to have my area of interest. And with that became an early advancement, and this one please here. Pardon me. There we go. Was the first advancement, which was a software advancement with this, and that was this automation of this spray and pray approach. A lot of this before was completely manual. You would sit in front of your microscope, you would do all of your thinnings, and it worked, but it was, you know, a time sink for research and researcher. So software was the first improvement to help with this, and you could set up as many sites as you want on as many cells on a grid, set your parameters, and walk away. So what we're looking at here is somewhere in the order of about 15, 20 lamellae that were done automatically. And then you bring it into your cryo TEM, and then you keep your fingers crossed that maybe one of these 20 does in fact contain my area of interest, my protein of interest, my interaction of interest. But that low yield was not always the best approach. Time on cryo TEMs is rather expensive as well. So the idea of incorporating what cell biologists have been doing for a long while into this workflow really started to become more and more appealing. I'm just going to dive past this here. And this is like that resultant process, by the way, of let's just hit all of our lamellae, our areas, let's create all these lamellae. So the more efficient approach for targeting included the incorporation of fluorescence inside that intermediate step, that Fib step, to allow you to actually say, well, if I could target, or if I could label something inside of a cell, then I should be able to target it. And it's proven to be a very nice, effective approach for the next step in tomography, which is fewer lamellae, but more meaningful impact on them. And we do this by incorporating a fluorescent microscope inside the chamber, which will allow you to turn on the fluorescence, assess, does my sample have the fluorescence? Where is it? And then this iterative process of ablation back to fluorescence. And you can confirm in, I think, a fairly intuitive way of how of the actual inclusion of your fluorescent area inside of the resultant lamellae. So a little bit on the fluorescence. So we are quite limited when you compare it against, you know, these high-end confocal microscopes, because we're working inside of a cryo microscope. We need to maintain a decent working distance so that the heat from our fluorescence doesn't transfer onto our samples and devitrify it. So as such, we found a happy medium of, you know, about a 20x, some samples up to 50x objectives to basically allow us to understand what's happening inside of our cells. I think for these techniques, what's going to actually be of equal importance, not just the objectives, but is our cameras, because we're not going to have the ability to switch around our objectives like you do in a typical light microscope setting. So a large camera is going to allow us to do this Google Maps style zoom in to really start to get a feel for what's happening, whether we're looking perinuclear, more off towards the edge of our cells, to give us that bit of information to say, here's where I want to be, here's what I want to target. And that workflow looks kind of like what you would imagine it to. So turn on your fluorescence. First, this is a micro-tracker example. It looks a little bit out of focus because, you know, we've done several Z steps through here. A max intensity image will define our area of interest. So in this case, you know, we want to target this little cluster here of mitochondria. Turning off the fluorescence, by the way, here's what we would say. You know, you don't have a feel for what's going on inside that cell. Alignment between fluorescence and your E-beam is actually pretty easy because a lot of these more modern fluorescence systems include a reflection mode. Reflection mode is, for all intents and purposes, another way of looking at your SEM image. So you can correlate your reflection against your fluorescence against your SEM to ensure that we're in the correct plane and our cells and our XY is dead on. From here, when we kind of turn on the eyes of the Fib, we start to get the feel for where we are relative to our E-beam and our I-beam. Then that process I just described of ablation, shuttle back, confirm can happen. I wouldn't recommend doing a cut and going back to your fluorescent every single time. It would just be a real big time sink, but you can get a good feel for where your fluorescence is, do some ablation, come back, assess, and then make your conclusion that, you know, if you start to see your fluorescence waning off the top of your lamella, you're probably right there at the surface. And then you could bring in the bottom part of your fibbing to thin down to about this 200 nanometers. And we could then confirm in our final lamella that our area of interest is in fact there. And some of these newer mapping softwares, which go across the platforms from Fib into our TEMs, can then read some of the metadata to help us get right back to these same areas. So we talked about how you went from one lamella to several lamellae in this blind fashion, from targeting an individual to or an individual site to the fluorescent area. The next step obviously would be some type of automated targeting. And there is a more recent paper that I think is still in the preprints from Liz Wright's lab, which did just this and addressed just that. This idea of when we turn on our fluorescence, do I need to be there to assess and confirm? You know, again, it's It's a fun process to watch, but it is a little bit of a time sink. So the idea of can I target became more and more important because it's great to get away, just let the machine run on its own. We have all these automated features. And sure enough, this approach has just been put into the preprints. And the idea behind Liz Wright's paper here, and there's a whole lot happening here, but is to use an additional step of a fiducial marker in the form of a fluorescing bead to basically allow us to align a targeted area, which they define as right here, against these beads. So the idea is, as I've mentioned before, we have this ability to do some optical sectioning with our wide field system. So you would define the most focused point of your fluorescence against the most focused point of your beams, and that angle against our fib beam will allow us to know where in our cell we want to be. Now, there is one manual step in here. So the typical path from cell to thin down lamella is three ion probe currents, a very rough ablation to do some rapid thinning, and then 2 finer polishings, one to get us to about 250, 300 nanometers, and a very low current to take us down there. These last 2 steps with these automated processes have now become completely automated with a high yield success. I'd be lying if I said it was 100% successful on these, but a lot of the work which I've seen done in our nanoports is around 50% success rate in a completely automated fashion from thick all the way down to thin. So with that, we kind of now want to take that next step backwards. So we went from protein on its own to a grid to a targeted protein inside of a cell. The next step in this workflow is always, well, what does it look like in the animal? Can I do this same process? To do that, though, we need to incorporate a different technique because for 2 reasons. One, we can't put tissues on grids. And secondly, the thickness of it just requires a different approach. And that is an approach which involves another intermediate, which is called Cryo Liftout. A lot of this work is done with tissues which have been high pressure frozen into a specialized holder called a planchette, which I'll show you actually in the next slide. But the idea behind it is essentially locate an area of interest, and this is a cartoon of a C. elegans, which will become relevant in a few slides, but to use a super cooled needle to basically remove this bulk and place it onto a receiving grid, which is called a half moon, and then from there do this thinning down process. Now, what is cool and also costly is that it's an effective approach, but this chunk here is probably a micron or 2. The ultimate lamella, as I probably said ad nauseum, is going to be somewhere in the range of 200 nanometers. So there's a lot of loss which does happen, which kind of kills the idea of these large animals and really understanding what's happening in context. So there's a paper, which I'll talk about in 2 slides, which approaches justice in a serial method. One quick thing I will point out here about these lamellae is that we do have the science is improving that we do have these dedicated holders that will allow you to lift out from our planchette and put onto these specialized holders here with these posts which allow us to weld on and do these tilted tomographies free of any obstructions. And here's a really nice example of it. This was a muscle-based sample where as you will see as we dive through the ablation, we start to really see what's happening with the interaction of nebulin and actin, very high resolution again. So this is a tissue sample that ultimately reached about 4.5 angstroms resolution. Wonderful approach, but again, quite a high loss from a tissue down to a 200 nanometer lamella. The more recent approach to come out of Plitzko's lab is a method of serial liftout. So that cartoon which I described before of our C. elegans was actually the same model organism they used in this approach, while this is a cartoon here. Clearly, they did this on a real animal, of course. But the idea is when you lift out, instead of moving it onto that thin down post, which we then again lose all this material, to take a different approach and basically weld our full animal onto an intermediate and using what amounts to essentially a traditional 200 mesh EM grid minus the horizontal or the vertical bars, we do a bunch of redepositions of our material using our ion beam. So in this case, all the loss that occurs from tissue through semi-thin down to thin is actually preserved. Now, there still is loss in here, but I don't want to say insignificant, but it's much less loss than typical approaches. So I don't know how well it's coming on your end, but you can kind of see these weldings here. What they're basically doing is using a redeposition method from the copper adhering onto your sample and then freeing up each individual section. So those of you who've done some forms of array tomography or something like that, serial section TEM, this is the same methodology, but now we're coupling it with the Cryo ET approach to yield quite high results over a large field of view. The last little bit, which I will throw in inclusion with this, and I kind of like to tie everything together here, is more recent approaches with Cryo Liftout does actually also incorporate internal fluorescence. So if your animal does have some type of fluorescence in there, we have this ability to actually assess in real time as we ablate away to confirm that our finalized thin down lamella does in fact contain our fluorescing region of interest. Let's see here. And I think if I move along here, just kind of moving over into summary here, the idea of the advancements of SPA into Cryo ET are obvious. Going from protein through cell and cell into organism is really into a tissue, is just broadening out our sample while maintaining a lot of the strengths of what SPA has introduced to us, namely angstroms level resolution on cells as well as tissues. Some of the advancements of technology in the form of internalized fluorescence is going to certainly allow us to do some targeted approaches. Our cellular biologist friends will certainly appreciate this. And then Liftout, I see Liftout as this growing interest as we move our organisms from cells into tissues as well. With that, I think I've actually gone pretty quickly through here. But I want to thank you all for your time and your attention. Certainly welcome any questions on this, and I'm glad to chat about this after the fact as well.
All right, well, should I give you this microphone, Geoff? Yeah. And then I'll take the other one.
Oh, thank you.
So we have our microphone runner if anybody has questions.
I think
And one was right behind you.
Thanks for the talk. I was wondering, in the lift out, how large is the needle and what's causing the adhesion between the needle and the sample section?
What's binding the sample in the needle?
Yeah.
So the needle itself is nanometers thin at its tip. So
What is it made of? The needle?
Tungsten. So there's two approaches. The traditional approach, and I didn't want to dive into that and really bore the heck out of everyone, but the original approach was to bring your needle to your area of interest and then pull in one of the gas injectors and basically weld with carbon or something like that your needle onto your sample. You would then remove it from the bulk, put it onto that intermediate, break the needle with your ion beam, and then there's a lot of gunk that comes with the gas injector. So you have to kind of mill through there, relocate your area, and then thin down. These newer approaches, and again, I'm glad to totally entertain this. I just didn't want to kind of dive into it in case it was outside the interest, but there's a newer approach, this GIS Free Approach, which I think this is too far. I think this is going to be like the next big improvement in Liftout. And what's basically happening is
GIS is the gas injector system.
Oh,
so that's
excuse me.
when you spray the
Sorry.
volatile organoplatinum, and that works great in semiconductor because you use an electron to condense it. But how we're doing it with the cryo weld, it just non-specifically binds everything that's cold. So GIS free means in the absence of the sprayed organometallic that he's talking about. So
Excuse me,
please continue.
thank you. But so this new approach basically uses a little bit of copper, which it's kind of like a sacrificial bit from your grid. So the first step is actually bring your needle down onto the grid and use a redeposition from your copper to create this intermediate here. From here, and now it's a really tough, I don't blame if you can't see it. This little area right here is that intermediate. So when you bring your needle down onto your area, as opposed to just snowing your sample with the gas injector, we're going to use our E-beam, or excuse me, our I-beam to actually deposit some of the copper onto your sample, which creates a strong enough weld to allow you to then free up your chunk with your ion beam. Nice advantages to that are you don't have to fight through the gunk initially. From a practical user point of view, you could then just snip away your material, which allows you to not run through so many needles quite as often. So and this is the approach that they took, if I can find that slide, with the Plitzko group. So there was the intermediate, which is welded onto there and allows us to really nicely go across all these areas. Sure.
Yeah, doing this with the GIS would have been, I think, impossible because if you weld one section, it would destroy all the neighboring sections, right?
Yeah,
By burying them under. Let me ask a question while I wait for other folks to raise their hands and the mic to get back to them. With the in situ fluorescence microscopy, there's also now a cryo FibSem, the Arctis, where the location of the light objective lens allows you to look at the sample without moving it somewhere else. Is real-time imaging achievable? And if so, what can you do differently in terms of targeting because of real-time imaging that you couldn't do when you had to move it back and forth?
So targeting will probably be a little more accurate. It actually has to do with the fact that we don't have a SEM stage in there, but a ComPu stage in there to help with the accuracy. Real-time imaging is absolutely achievable. There's still the process of, turning on your I-beam in your fluorescence, but it's definitely going to make it more efficient in terms of your accuracy. Again, with that stage, the objective's a little bit better, so you're going to have a little bit more clarity. The camera is basically the same principle of the IFLM camera, but real time is 100% achievable with it. So it's this triple point solution. So I probably jumped over that part too. The IFLM solutions on a lot of the fibs today, it's inside your chamber, but off axis. So to go from imaging with your E-beam and ablating with your I-beam, you have to shuttle to your fluorescence. This newer technology, all three at a coincident point. So there is no shuttling involved there at all. So it's real time. It's a lot more rapid. The Arctis is like the Lamellae machine on steroids.
Thank you. I'm not a cryo electron microscopist, so this is a naive question. I noticed you mentioned that you mill the grid as well as the sample. So which I find interesting. So the sample is basically being bridged. The
Correct.
Grid is basically on the sides. The sample is a bridge that you're looking through. Is there a preference for grid material for this technique, or is this applicable to any material, or is there a preference? Do some yield better results?
I think a lot of there's specialized quantifoil grids, which was a lot of people were using for the SPA approach. I know that your first qualification will be you probably want a gold grid if you're growing your cells live, so that nickel or the copper grids are toxic towards your cells if you're growing your cells on there. I've seen some of the work done with more traditional EM grids. I think these quantifoil grids are just so much more supportive nonetheless, that when you're bringing it into your TEM and you want the most stable substrate, that the firmer grounding of a quantifoil grid is probably still the more preferred one. But again, I've seen some people do 100 mesh grids, like traditional TEM grids, and the data looks pretty dang good as well. So to your question, yeah, so you're milling it away, so you're losing that below substrate, but it's still anchored in on both sides. So
Chip, if I may, because I think it's the idea of the forces and the structural integrity underlies part of what Greg is asking about. Could you introduce the concept of the stress relief cuts?
oh yeah, absolutely. So, and I don't know if any of the images I have it, but what I could probably is just, oh, here you go, here, this will even suffice. So when we set up our patterning to go from cell down to lamella, the resultant lamella, if it was to be right here, as you point out, is anchored by the edges of the cells, but it does create quite a bit of stress on our lamella. So more recently, and you can kind of see it depicted here with these blue lines, the idea of adding in a stress relief area to kind of kill some of the tension on our lamella is almost standardized in a lot of these processes to basically prevent any buckling or breaking of our lamellae in the process.
Interesting. So, and when you tilt it, does there have to be a you want to measure the way at a certain axis when
Yes.
you do you come into at high angles some
Yes. So
interference from edge
in a
edge effect?
perfect world, you'll be 90 degrees in your tilting angle from your milling angle. So there's a lot of you know, I'd call them techniques, but basically what a lot of people are doing is taking like a Sharpie dot, putting it onto their grid before any milling. So when you move it from your fib into your TEM, you can approximately align it, but there's always going to be a couple of degrees of error. So the newer microscope, which Jason's referring to, this ArcGIS, actually took this and ran with it. So there's a newer grid which will be released with these ArcGIS tools as they start to roll out, called a Tomo grid. So it's basically a normal grid, but with wings on both sides so that you can only put it into its cassette in one way. Technically two, I guess you do 180 degrees. But when you, it ensures that your fib milling is happening 90 degrees to what your tilt angle will be so that you can achieve these higher tilts of up to 70 degrees, I guess, in the best case scenario. But yeah, so you absolutely want to try to make sure that your milling is, you keep in mind your tilting angle relative to your milling angle to give those higher degree tilts.
Thank you.
Oh, wait.
I'll run this one to you.
Okay, so you said that in an ideal session with automation, you can get as many as 15 lamellae. So how do you battle ice contamination in this case? Is there any other ways than to go back and manually polish all of them one last time?
So there's two answers. One is better, a lot of the companies have thought about this and are improving vacuums to reduce contamination rates down to a few nanometers an hour. So obviously you want to be as efficient as possible to kind of counter what you're talking about. Certainly good polishing methods are going to have to come into it. So if you're cutting, 15, 20 lamellae in a single session, then I probably wouldn't polish the first one and do everything in an individual fashion. I'd probably go iteratively and polish everything at the absolute end. No perfect answer to that. But these newer solutions, particularly the ArcGIS system, again, it's actually an autoloader based tool. So we're only exposing one grid at a time to our environment. The rest of them sit in clean nitrogen, so they're not building up any contamination as they go.
Let's take Wei and then the online comment.
Geoff, really nice talk. So I'm really interested in the Cryo Liftout. So can you talk a little bit more about that particular slide? So how did you prepare C. elegans in that one? And then what is, how big is your each block? How many blocks you were able to get? And then overall, what is the yield for that? Lots of questions.
I just want to find the slides. I could talk my way through it. I think it's the other way, actually.
The serial Liftout.
Yeah.
The C. elegans.
Wait, no, this is her, so it's the other way. Excuse me. So the sample was high pressure frozen using waffle method to minimize the amount of thickness through the high pressure process. They then, didn't actually go too far. So I'm sorry. So the rest of the questions were how did they number? So, and I didn't want to put too many. So the rest of the paper has another figure with it, which shows they have about 28 serially laid down in this fashion with minimal loss. So I don't remember if they defined the minimal loss. I can look back on it, but I'd imagine through the polishing process, they're taking away, you know, tens of nanometers, if not maybe half a micron. But it was done sequentially, so there was no loss in between the laydown. I just don't know if they went end to end on the worm. I don't think they really defined it in there. But you know, this redeposition method was key to it because essentially you're putting it down, welding, breaking, repeating that process iteratively throughout it.
So how I forgot, I should probably know this. So for C. elegans, how long is it? You basically go along as
Yeah, well,
once.
grown C. elegans can be up to 1 millimeter. I think they used younger C. elegans, so it didn't quite get that long. So, and then the diameter of them is somewhere around, I think, like a few hundred microns, like 400 or 500 microns, I believe. So it certainly can fit easily inside the width of 2 grid bars.
So each is approximately, if you say this is a tomography grid and you just remove the kind of vertical bars, it should be about 100 micron in size. Is that
This is
right?
100 mesh micron or bars, so I think the distances are even lessened. But I mean, I gather your point. Like, it sounds like there's a lot more gap than the width of it.
Right.
I'd have to dive back into it to really understand the geometry of the grids.
So if it's 100 mesh, that'd be 254 micron
Spacing.
center distance on the bars.
Right.
Yeah, so I'd have to look back on that one to really come to terms with the sizes.
Okay, thanks.
Sure, fair question.
Okay, let's go to the online question, which was in the process of IFLM, in the fluorescence microscopy, is there a fluorophore bleaching issue on the FIB process? And if so, how do you resolve it?
On the FIB process, so is the FIB affecting the fluorescence? I've not seen that one. I know that fluorescence is a lot more resilient under cryo than as opposed to wet because you're not oxidizing, bleaching it at that same rate. I've not seen the FIB actually kill fluorescence. I'd presume that anything, if you expose it to an I-beam long enough, probably will affect the conformation of a GFP or something like that, but I've not seen loss in fluorescence from the FIB.
So if there were, it would be dependent on the penetration depth of the ions. So could you speak a bit to how deep from the surface does the FIB actually affect?
Sure. So a couple of papers that came out beginning of 2023 assess just that question. And I know that the final polishing for gallium was about 60 nanometers from the top edge or bottom edge, showed some type of depletion into your lamellae. So they used ribosomes as their model, and they found that once you got below 60 nanometers in your lamella, any resultant loss from FIB beam effect was completely diminished. They did the same test actually with plasma, independent groups. They weren't comparing the methods, and plasma was about half of that. So about 30 nanometers from each surface showed effect from the FIB beam. And beyond that, we were fine. And they also used ribosome as their model.
Okay, so even in the worst case, if the FIB completely destroyed fluorescence, it would only do so 60 nanometers from each side. And as far as the fluorophores and the bleaching, I've heard tell that a lot of the sources of fluorescence actually behave differently at cryogenic temperatures than at room temperature. Is that something that you can speak to?
Not that I've seen, but I know what I see in front of me. I'd be curious to dive more in and see what the, two conditions yield, but not that I've seen. Again, I don't know everything yet, but.
Well, we do have time for one more question if anybody has a last one, either online or in person. We got one.
Thanks a lot for the very rich introduction. I was just wondering, maybe I missed this. What's the thickness of the milled lamella in the FIB process? And if the thickness itself will affect like which angle we are actually milling in a real cell that affects if we are looking at a specific structure of an organelle inside the cell. Yeah.
So thickness can vary, obviously. Like I said, probably ad nauseum. 200 is the one that people target in an automated fashion. Oh, yes, 9 meters. Obviously, in an automated fashion, there's going to be a little bit of error in there, depending on your ice, how much sample is there as well. So when you see a resultant bar, there's usually a pretty big error bar in there. Angle, I suppose it probably could affect it. I've not seen anything that, if you're milling at like 12 degrees versus 8 degrees, you're going to be that much more thin or thick. Because at the end of the day, I suppose changing your angle will just mean the amount of material that you're going through. I'd imagine the more oblique ones, you're probably traveling further through a cell than a more, higher angle one. But I would imagine that you'd be pretty close regardless of your angle.
Thank you so much.
No, thanks. That's a great question.
Well, questions now being over, as with all of our visiting speakers, we'd like to present you with a little memento of the Institute for Quantitative Biomedicine.
Great.
So please go ahead and open it up.
That's awesome. Yeah. My birthday's next month, but thank you.
Well, happy early birthday.
Thank you all. This is great.
Thanks, everyone. And for the students, we'll go have lunch with the speaker and get into some more discussion.
All right.